you, Ramati. Thank you very much for the opportunity to speak here. My name is Christoph Goebl. And I would like to introduce myself. Before I came to New Zealand, I worked in Canada in a cancer lab. And there we discovered new cancer drugs. So we went in that lab from identifying a new target to developing a drug to going into clinical trials, stage 1, stage 2, and then commercialize that product. And I have to say it was really inspiring to see the whole process there. I think those things are possible. I know they're challenging, but that's exactly, I think, why we are in this job and we want to achieve something. When I was asked to give a talk about our project that we perform in the ISS, I really couldn't resist to show you this experiment here. It's a NASA twin study. It's a very famous study. It's now about 5 years ago that it was published. But in 2015, NASA took a twin couple. They sent one of the twins to the International Space Station. They kept the other person on the ground. And they measured data on them. They measured health data on them. Because they were identical twins, they could nicely compare the results of that. And it was really impressive. This study has sparked a lot of interest in human space travel because we learned so much about health parameters. And because we talked today already about giving back to communities, and we heard about the New Zealand setup, at the Aotearoa setup, where we have a lot of rural communities, I would like to point out that through these studies, and because the ISS is a limited space very, very far up there, a lot of medical procedures, a lot of ways we measure health parameters had to be miniaturized. So there are lots of engineers have thought about how can we make those measurements cheap, how can we make them serviceable very easily, how can we make them accessible for non-specialists? Because people at the ISS, when they measure some health parameters, they are not doctors, there are no nurses there. The data has to be understood by people out of the field. So I would like really to think of the ISS research to really spark a lot of those translations, making very complicated procedures available on ground, making them available for rural hospitals and rural setups. I really like the idea of saying this is basically research that is very, very far away, but it's really very useful on the ground. Now, one last comment I would like to make before I start talking about my own research. I don't really care about billionaires in research. I care more about the people here in Aotearoa, the whānau, and I care more about the 8 billion people here on the ground. But because we can learn so much, I really like the idea of generating that data and being surprised by what we find out. Now I give a bit of a lesson. I didn't know you all know about P53, so I started talking about another protein. I'll talk about the protein called P16. And maybe that's for Matti, so that's your personal presentation for you now. So yes, great.
Thank you.
I would like to mention at first how complicated of a disease cancer is. When we say cancer, we really talk about an umbrella term. So cancer consists of hundreds of different types of cancers. And those hundreds of different cancers also have different subtypes. It's a large number of diseases. We don't have a pan treatment. We don't have a single treatment against cancer. Every single cancer needs to be understood on the cellular level. We need to understand the cellular pathways that are happening there. And then we can make a very informed decision on what exactly we want to treat. And we need a very specific drug that interferes with those specific pathways. It's a very, very complicated disease, and we are only really just halfway there getting our hand on potential treatments. So I would like to show you on the right, this is a very famous graph, and I just heard before, some people know that graph already. Doug Hanahan has published this. It's called the hallmarks of cancer. So you can see here now different cellular pathways contributing to the development of cancer. It's a complex interplay between a lot of different pathways. And depending on which cell type we're looking at, it's a very, very complex to solve. And we need to understand most of those aspects in order to find a new vulnerability and how can we address new questions in cancer. So I mentioned already that we have hundreds of different types of cancers. We have about 10,000 different types of proteins in every single cell. And because cancer is a genetic disease that has individual mutations that basically derail the healthy cell, that transfers it into a cancer cell, we have to deal with millions of mutations. It's a very, very complex disease. Now, I would like to introduce a very specific paradigm here. We didn't hear about paradigms today. I would like to talk about paradigms today, and I also like to talk about breaking those paradigms. On the left, you see the 3 Nobel laureates from last year, David Baker, Demis Hassabis, and John Jumper. And these guys have done something incredible. I spent 4 years in my PhD to solve one protein structure. You've seen a protein structure before already. To generate such a protein structure, solve it experimentally, it took me 4 years. I had to live in the US for 1.5 years. It was quite fun at that time because Obama was president and it was a very motivating period. Everybody loved it at that time. But what these guys did now, they came up with with an AI tool. So they wrote a very smart code. They used artificial intelligence to learn about protein structures. They used those structures as input. They generated a software that can predict structures. And they solved hundreds of million structures in basically 2 or 3 years. So it's an incredible progress that we made there. And for me as a researcher, the time before AI-based structure prediction and the time after, that's 2 completely different timescales. We do research completely different nowadays because we have access to all those structures. Now, one thing that underlies this structure prediction is this sequence structure function paradigm. So you can see here on the left, so you've got a protein sequence and it's consisting of individual amino acids, like the building blocks. And that sequence will be translated into a single structure, which then is associated with the function. And this sequence structure paradigm is something that has been established over the last decades. We've got 50, 60, 70 years of solid research that tells us a single protein sequence has a single structure, and that single structure has a specific function. Now here is where a cancer protein comes into play that we research a lot. So this protein is called P16. So in contrast to P53 before, this one is called P16. So the ones that are called P something are usually proteins that have been discovered very, very early. So they must be quite important at least. That's what we think. And what you can see here now in P16 here is that this is the structural model that we generated. It looks the same if you solve it experimentally or with the computer. The data is so good nowadays we can't distinguish that anymore. And what P16 does is it inhibits cell division. So it's a very, very important protein. If P16 is present, the cell can't divide. And that means a cancer can't form either. So it's a very, very important protein to basically keep cells from dividing. Now, this protein is one of the #1 enemies for a cancer because what a cancer wants to do is the cancer wants to spread, the cancer wants to divide all the time. So that means that in most of the cancers we find that P16 is modified. Now, what we discovered now, we looked into this protein now and we thought about this paradigm that this protein has a specific structure, and we found that by a very small modification, a single oxidation event, really just one electron that changes basically on the protein, leads to a complete rearrangement of the structure, and we get a completely new structure. And what the structure on the right that you see here is now an electron micrograph. So you can see here from electron microscopy that the protein forms these large aggregates, and we call those amyloids. We know them from neurodegenerative diseases. They are very common, amyloid beta in Alzheimer's disease, or we also have Parkinson's disease that is based on those proteins that form those amyloids. So we found that P16 can also form those amyloids. And something that was really surprising to us is that if we reverse that oxidation, you can see here we call it reduction, this chemical process, it goes back into that monomeric state. So this protein does not have one structure, it has 2 structures. And that is something we were not aware before. We really thought, and this is, I think you talk to 1,000 scientists and you tell them about your protein, they will ask you, what does the structure of that protein look like? None of them will ask you, what do the structures of this protein look like? Because that you can have multiple structures for a single protein, that is really something that we didn't have conceptualized yet. There's a growing field now. It's not the first example, it's the first dramatic example because a single oxidation event causes this transition. But we learn now more and more how those proteins form those tangles and those aggregates. And that usually causes also another function for the protein. So this is really expanding the knowledge of our protein understanding. And more importantly, what we found is that once these amyloids are formed, once P16 is in this state here, it basically promotes cell division. It is no longer active. It no longer inhibits cell division as it does in its native function, and it completely changes its function too. So this is the protein we are studying, and we think we can really understand some of the cancers a bit better that way. Now here's another example of why P16 is a protein that is really, really interesting. You can see here 2 mice. They're littermates. They're two sisters. They're both female mice. They have been genetically modified to a small extent. On the left, you can see the mouse that is about 3.5 years old. And that's what you get. The life expectancy of such a mouse is about 3.5 years. You can see after 3.5 years, it's a senior mouse. You can see the fur is a bit missing, it's losing a bit of the hair, it's a bit wrinkly, and that's what you get. The mouse is aging, and at some point the mouse will die of a natural cause. Whereas the mouse on the right is at the same age. There's no difference whatsoever. The only thing is that when the mouse produces a lot of P16, which naturally happens because it is driving the aging process, this mouse here, when the P16 is produced, the levels will go down, so it is genetically modified to not allow too much P16. And you can see, although it is in the same age and has been treated in the same way, got the same food, it looks way, way healthier. And when we measure those results, it is way healthier. So this mouse can grip stronger, can swim faster, can memorize a maze where it finds the treat in one of the corners way, way better than the mouse on the left. So basically, we know that P16 has multiple functions. It is suppressing cancer, and at the same time, it is an important aging protein. And that really shows how complex proteins are, because you want enough P16 to suppress the cancer, but you don't want so much P16 that you drive the aging process. So it's not a one-dimensional thing. You say, oh, P16 is a good protein and it protects us from cancer. If you have too much P16, it drives the aging process really, really rapidly. It's a strong phenotype. We can see that. And that's not just true in mice. We also found that in people. So this protein is central to a lot of pathways that we know are at the core of a lot of cancers that are forming. Now, we were very, very excited that we could perform now some experiments with Associate Professor Sarah Kessens, and Sarah is here and she will join us for the panel discussion later on. So what Sarah did is she managed to send proteins into space, and at some point she knocked on our door and she said, do we have an interesting protein? And I think I said yes at that time. I was maybe a bit overconfident, but it was really interesting for us to be able to send the protein to the ISS. Now, the hypothesis is here that when we have this protein that we know can switch its structure via oxidation, the question we ask is the radiation in space enough? Is the radiation in the ISS enough to induce this transition? So the ISS is well shielded, of course, because you want to protect the astronauts, but still it is more radiation up there than we have here in the ground, where we don't have the atmosphere that protects us. So we sent this protein up there, and you can see here some pictures. I would like to mention Shelby, and Shelby should be here. Oh yeah, thank you very much. Shelby is the student basically that was working tirelessly to make that happen. I think we got a deadline of about 3 months, and I don't think you had a free weekend in that period of time. Many nights, many emails late at night, I remember that. But you powered through, and thank you very much, Shelby, for delivering. In the end, what Shelby did was she generated now a lot of different samples of P16 that went to the ISS. We generated the ground control samples. We produced those chips with some engineers that we collaborated with in order to store the protein the way we wanted to, so that there's no real shaking that can change any structural properties of the protein. So this is a very, very sophisticated setup, and we all could do that in a couple of weeks because we didn't have too much time to plan it. And you can see here Sarah in the US at the International Space Station Center, at the Kennedy Center in Florida. You can see here Sarah setting up our samples, and then you can see here the countdown to the start, and this was the SPX-30 mission that transported our samples into the ISS. And I remember we did a launch party. I called it a launch party because I think it was the only party I accessed in the last 20 years, I think. It's a bit sad. But we had pizza. It was pretty much around noon at our time. I think it was Friday, 11 o'clock or something like this. And it was really exciting. The whole lab was sitting there. Most of us were working on this protein at that time. We were watching the rocket taking off. We were hoping that nothing happens, of course, because there was a lot of work also from our side working into this. And the protein stayed now at the International Space Station. It was supposed to be there for 30 days, but then the space weather wasn't good, so they couldn't bring the sample back the way they wanted it. So we had to wait another 10 days, if I remember. So the sample was at the ISS for about 40 days now. And then we got it back. I remember Shelby flying to the US, coming back, very exhausted, handing over the protein at the airport. Somebody from our team picked the samples up, and it took us 2 days, I think, again, day and night, to analyze the samples right away. And what we found was pretty much what we were thinking we might see. So on the left, the ground control samples were stable, they were monomeric, they were behaving in a way that we always thought P16 would look like, but the samples from the ISS, they were actually forming amyloids. Now, one thing I would like to quickly mention is that we would love to have the structure of those amyloids. AlphaFold cannot give us the structure of those amyloids. AlphaFold is written for this under this paradigm. So you go to AlphaFold, you click on the protein, you get one structure there. You can't get an alternative structure because the software doesn't know about that. So we would love to know the experimental structure of these amyloids. And we spent a couple of thousand hours. I think I was calculating yesterday. I think it took us about 1,000 to 2,000 hours to try to solve the structure. We couldn't do it. But because now of these ISS samples that we could get back, and we got this amyloid formation there, we're now really interested in thinking about how we can apply radiation, and in this case, this is radiation therapy for cancer patients, what the radiation therapy does to our protein. Because we know that when the cancer is treated, that the peripheral cells also get damaged, and we think that P16 there can form those amyloids. And it's very well known, actually, those peripheral cells are very proliferative. They can grow very quickly afterwards because they get a lot of damage. And so far, there was a lot of research on DNA damage, but not so much research on protein damage. And we think, in this case, where we switch the structure of the protein, that's actually quite an interesting case to investigate. And here's the last thing I want to show you. When we looked at our ISS samples, we were very surprised to see that instead of having those really not good-looking amyloids that are very hard to analyze, we found very, very straight needle-type amyloids, and those are very well suited for structure determination. So we are currently collaborating with a lab in Australia, and we think we can solve the structure from that. So that was something we didn't expect at all. Just got our package back, and as a surprise, we got very, very nice, strong amyloids that we saw there. Yes, thank you very much. I think I ran a little bit over time now. I apologize for that. But of course, I want to thank Shelby again for all her work. And there was also Pierre and Emily involved. Dr. Wesson Morris from the University of Canterbury is a strong collaborator. The Martha Herorda Center, where I'm working in Christchurch. And then, of course, Sarah Kessens for all her help with this mission and our funding bodies here. Thank you very much.